Okay.
And we can let people start to come in. So, right.
I received a couple of emails from people that they didn't get the link details, even though I send automatic emails. So I keep that in mind in case we have some troubles in between. I will just jump to the MailChimp and send them again, just in case.
Okay. Yeah, that could be good. All right. So yeah, I can say a few words, and then I'll turn it over to you, Effie, and you can do the introductions. So sound good? Okay.
Yeah.
Okay. Well, as folks come in, just to kind of show you here, we have up the advertisement that hopefully some people have seen. If any has not, hopefully this is exciting news, that the Timing Research Forum is going to be doing their third conference or meeting, and it's going to be hosted by Joe Patton at the Champalimad Center for the Unknown in Lisbon, Portugal. And this is going to be happening in October of this year, so October 4th, 5th, and 6th, to make sure to mark on your calendars. There's not a lot of information right now. We do have plans to have a website up for this, as well as more details. We will be asking for submissions for posters and talks and symposia. That's likely going to be happening sometime around May is when we're going to be looking to receive those and review them. So we're very excited, and please stay tuned for more information as it comes in, and we'll have more details for you soon. So Effie, I will pass it over to you now.
So welcome everybody to the first Journal Club of the year. So even though it's late January, I would like to say Happy New Year to everyone with health. And we are very excited to host today two very talented researchers. As usual, we would like to have your microphones off. We will have 20 minutes for each talk, and then you can place your questions in the Q&A section or raise your virtual hand to ask questions. And also we would like to thank Dr. Weiner, Dr. Vatakis, Dr. Teki, and the ACR committee for putting these meetings in place. I will go on and start with our first speaker. Her name is Saeedeh Sadeghi from Cornell University in New York. She is a PhD candidate at Cornell University at the Department of Psychology and Human Neuroscience Institute. She has a bachelor's degree in computer science and 3 master's degrees, 1 in machine intelligence from University of Tehran and 2 master's degrees in system engineering and psychology from Cornell University. At the moment, she works as senior researcher at the Affect and Cognition Lab at Cornell, and her interests include time perception, heart-brain interaction, interoception, and emotion. And her work on the perception of time in an immersive VR environment has recently featured in multiple news channels, including the Tech Explorer and the International Business Time. Today, she's going to talk about subjective time and interaction with heart rates. So, Saeedeh, the floor is yours.
Thank you so much for the introduction. It's a pleasure to be here. Thanks for organizing this event. So my talk is about the interaction between time perception and heart rate. I think everyone here knows that subjective time is malleable, that subjective time does not equal objective time. For example, this man who is running here, chasing the train, for him, time feels like it's slowed down. And the movie industry actually uses this effect a lot to induce that a moment is intense by making it slow motion. So in this talk, specifically, I'm focused on duration perception. What I mean by time perception is perceiving a interval or a stimulus that has an onset and an offset, and there's a duration in between, and person's perception of this duration. So there are factors that are known to systematically lengthen our perception of duration. For example, attention. When we attend to something, it feels longer. Or arousal, especially unpleasant arousal. For example, when we see an angry face or a spider, that in the time that we look at these images feels longer than seeing a neutral stimulus. So why am I studying time and heart relationship? Why does it matter to me, at least? The thing that motivated me was that, well, all we have in life is time. Like, our life is composed of time. Is like the entire duration of life, or the moments that we experience, is like the container is time. And to me, it was fascinating, the idea that something physical, like heart, an organ in our body, heart that pumps blood, it can alter the experience of our life, like our duration, our perception of moments of time can be influenced by something that we have no control over, which is like it's controlled by the autonomic nervous system unconsciously. So that was fascinating to me, and that motivated me to study this. The classical way of thinking about the relationship between heart and time is that, we know that subjective arousal lengthens perception of duration. When we are aroused, time feels longer. There is kind of good amount of evidence supporting this. And we know that subjective arousal also increases the heart rate. So people with this logic said, well, maybe higher heart rate is related to lengthened perception of duration. And this was especially appealing because heart has beats which resemble pacemaker. It's like there is an internal clock in our chest. But the evidence on a direct relationship between heart rate and time perception has been not that strong. There are a lot of inconsistencies. And this is actually where I started, that, okay, maybe I try something similar or a little different from other people did, but like just test if there is a direct relationship between these two, between heart rate and time perception. And at the time that I did this project, I was in engineering school. And my advisor was interested in experience of passengers on public transportation. So we compromised to work on time perception on subway trips. So it was supra-minute time scale with realistic stimuli that people experienced in a virtual environment. They experienced subway trips. They experienced 5 subway trips with 5 different crowding levels in a random order. And we recorded their heart rate as they were experiencing the trip. So this is an example of the experience of crowded condition.
Stand clear of the closing doors, please.
So they experienced these kind of trips. Each trip was between 1 to 2 minutes. And after each trip, we asked the subject how long was it in the number of seconds, just estimate the duration, and how pleasant was it on a scale of 1 to 7. So the behavioral results were were actually, as we expected, very neatly followed our hypothesis that crowding induced negative feelings and it lengthened perception of travel time. So when the feelings were more negative, they estimated the travel time as longer. But the heart rate results were not that neat. So we estimated the average heart rate and tested if it's correlated with crowding level, and it wasn't. Well, we expected when the crowding is higher, heart rate would be higher, but it wasn't the case, significantly not effect. And we also tested with subjective ratings of feeling about the trip, and there was no correlation. And we also tested with perceived time, and there was again no correlation. We also estimated heart rate variability during the trip, which is like how much heart changes during the trip, or the slope of overall slope of change in heart rate during the trip. There was like a weak effect of the slope, but overall we didn't see anything strong with all these different tests, statistical tests. And it wasn't too surprising given the previous literature that mainly suggested that it's subjective arousal that is linked to time perception rather than heart rate. But why did we fail? What's wrong with this way of thinking? Well, arousal should increase heart rate, and heart rate should increase duration, perceived duration. So maybe we should revisit this assumption that heart rate is arousal. So yes, it is true that heart rate, when we are more aroused, when we are anxious, for example, our heart rate is overall higher. It's like heart rate here is illustrating our state, our emotional state of being anxious, or when we are bored and we are about to sleep, our heart rate goes down. But on the other hand, also heart rate responds to environmental demands, to stimuli, different stimuli. So there are two main types of reaction: defense and orienting. So when there is a intense, very threatening stimulus, like if I see a snake right now in front of me, it will trigger a defense response, which increases the heart rate. Or if I see a stimulus that is not too much intense, but it's relevant to me, it's novel, or it's unpredictable in some ways, it attracts my attention, then my heart rate goes down. And that's the orienting that is part of the orienting of attention, this deceleration of the heart rate. And this relationship is not just from the brain controlling the heart, which I just talked about, like the brain perceives a stimulus and decides that the heart rate should go up or down. The brain doesn't do this just for fun. There is a reason for this change, and that is because heart rate also influences the brain and the body. So when heart rate goes higher, from lower heart rates to higher heart rates, the metabolism in the brain and body increases, and it increases vigilance and speed of actions, of the preparedness for action. And that explains why the heart rate increases when the body wants to go into the fight or flight mode, when we need to be fast and act. On the other hand, when heart rate is lower, the sensory intake is easier. So when we need to be passive and just perceive things from the outside world, that's what happens when we need to attend to something. Heartbeats are a noise to the cortex. We don't need the information from the body in that moment. We need to just get things from the outside world. So that kind of explains why heart rate goes down when we attend. And it's not just the orienting is not just for a transient seeing a stimulus for a short time, but even in a prolonged durations, when we attend to something, our heart rate on average goes down. So I thought that in a long time scale that has many beats in it, like tens of seconds, it's impossible to separate different sources of changes in heart rate from each other and different ways they interact with the brain and with time perception. So in my next project, I actually changed my major to psychology here. And my next project was studying the very far end of the spectrum of studying subsecond stimuli, how they interact with heart rate, the perception of subsecond stimuli. And in the subsecond range, I actually went way extreme to shorter than 200 milliseconds, which is shorter than a single heartbeat, much shorter, like it's a quarter of a single heartbeat. So we had a temporal bisection task. The stimuli were white noise tones in the range of 80 to 188 milliseconds. When the subject wanted to do the task, there was a practice session that they hear the 80 millisecond tone, and we said, well, this is short, and this is what they hear. This is short. And then they hear the longer tone at 188, and we said, this is long. And then they after they learn to associate these durations to short and long, in each trial of the task, they hear the tone that was between short and long, like this, and they had to tell if it was closer to short or closer to long. So it was a 2 alternative choice, short or long. And there were 210 trials. Each trial, the stimulus was something between short and long. There were 5 levels of duration between short and long. And we recorded the heart rate as they were doing the task. So there were 45 subjects in this task. Now let's see what we expect to happen to the heart when the subject is doing this task. So this is the ECG signal. So the peaks of these peaks are called RR. The peaks are peaks of the ECG, and the distance between them are RR. This is the interval. So when the tone is played, the thing is that responding to tone is motivationally relevant. Well, the tone is not too intense to cause a defense, but it is expected to cause attentional orienting after its onset. So if we plot the RR signal, the changes in RR, so RR increases, which means the heart rate decreases after the tone onset. And this is what happens. This is the average of all trials of all subjects, the RR signal over time in any trial. So time 0 is the tone onset. And we see that the RR goes up and reaches a peak and then goes down and before the next trial. So this is the what we call cardiac orienting response, or core. Then we looked at the subjective temporal biases and how they interact with this core, with this RR signal. So we divided the trials into dilation and contraction. Dilation is when the stimulus is short but is perceived long. Contraction is when the stimulus is long but is perceived short. So time is contracted. And we plotted the RR signal for all average across all contraction trials versus all dilation trials. And what we see is a significant difference that when there is a dilation, this dashed line, the core, the orienting response is stronger. The heart rate changes more. And this can be explained by attention effect. We know that attention is related to lengthening of subjective time. So that explains why when the core is larger, time is more dilated. We also looked at temporal consistency. So we combined the dilation and contraction trials and called them inconsistent. And we also called when the response was aligned, was the same as the stimulus, we called it consistent. And we compared the inconsistent trials with consistent trials in how the heart rate dynamics is during the trial. So on average, the inconsistent trials, this dashed line is larger and a bit shifted to the right. It's like shifted to the up and right. So RR is on average larger when it is inconsistent, which means heart rate is overall lower when it is inconsistent. So a higher heart rate is related to more consistency, and that can be explained by higher vigilance. So this was also correlated with faster response time. When the heart rate was lower, the response time was faster, the consistency was higher. So it's more vigilance, more preparedness to act. So we went further from just these plots and regression and analysis, and we thought, okay, if heart rate has something to do with perception of time, it can be embedded into the model, into the decision-making process, when we accumulate evidence to come up with a response in a single trial. Heart rate dynamics might have something to do there. So we did the cardiac drift, the drift-diffusion modeling, which models how the brain comes up with a response in a trial, which is like that there is some evidence is accumulated over time. We start from an initial point, some evidence is accumulated until we reach a threshold, and that is response time, and that's when we decide, depending on which threshold is hit, we decide if the response is short or long. So this model can be fit to response time and choice data. It fits to behavioral data, and it has some parameters, starting point, the decision threshold, the drift rate, and the non-decision time. I'm not going too much into detail because of you can read more details about it in our paper, but we also embedded the cardiac dynamics into model parameters. First, we had a base DDM, and then we had a cardiac DDM, which was had heart rate dynamics into the model. So remember, this was the core, the orienting response following a stimulus. We extracted two features from it in each trial: the pre-RR, RR before the stimulus, and core, the amount of change in RR following the stimulus. And we set the model parameters, Z, V, T, R. These are the initial point, drift rate, and non-decision time components. We set these parameters of the model to be a function, a linear function of these cardiac measures that we extracted for each trial. So first of all, let me say that the way we set them to be a function of these measures was based on some hypothesis about the effect of these measures on sensory intake, on vigilance, on speed. And the results, so we found that the cardiac DDM fit to the data better than the base DDM. So it explained behavior better when we considered heart rate changes in the decision-making process, like embedded in the decision-making process. We could explain response better. And the effect of the cardiac terms were significant. Heart rate dynamics influenced encoding of the stimulus and the rate of evidence accumulation. You can read more of this, of the details of the model in our manuscript, which was accepted last week in psychophysiology. So it should be out in a few weeks. So back to the main question, does heart rate interact with subjective time? We did two projects on two ends of the spectrum of time scales, shorter than 200 milliseconds and supra-minute, longer than 1 minute. So in project one, that was longer than minutes, we found weak or no correlation between heart rate and time perception. And well, one way to think about it, I think if there is one take-home message from this talk is that heart rate is more than average. Average heart rate or heart rate in general is more than arousal. There is a lot into heart rate than just arousal. And for the sub-second duration, we found multiple concurrent relationships between heart rate and time perception, and our modeling showed that heart rate is like entangled in the moment-to-moment temporal perceptual process. So I don't have much time, just 1 minute. There is something I really have feelings about that. That some people, well, cognitive neuroscientists are basically a little biased about when they think about heart versus the brain, or in general, the peripheral nervous system versus the brain, that some people think that heart rate is less informative than neural correlate, that, well, we need to find a neural mechanism in the brain. Like you find some correlations in the heart rate, what about the brain? If heart has a bidirectional link with the brain, I think it's not a good way of thinking about that. Like we need to find mechanisms in the brain. Heart can be thought of as a node in the network. We know that everything is with network. Like there is no single time region probably, and heart could be part of that network of places that give rise to the sense of time. If there is a bidirectional link, then we can think of heart as a correlate of subjective experience. Why not? Why do we have a bias against, a resistance against thinking of heart or any other bodily organ as part, like as same as a brain region, like as part of the what gives rise to that subjective experience? Thank you for your time and attention. I would be happy to answer any questions.
Thank you so much for your presentation. That's a really interesting topic, and I actually like the question that you raised at the end, or the thought that you raised at the end. I will go through the chat now to see if there are any questions there, and then I would like to share my thoughts. It's not a question; it's just more like a thought.
So I have a problem of I need to stop share to see the stuff going on. Okay.
Okay.
Martin, the question.
Yeah, that was a great talk. But is it okay if I ask the question?
Yeah, of course. Go for it.
Great. Great talk, Saeedeh. Thank you. And congrats on getting the paper published in Psychophysiology. I'll read it with interest. I just had a question about the DDM model that you used. It was a little brief, but it looked like you varied drift rates, but you also had non-decision time and bias as well. So the first question, did you see any effective bias, like the Z parameter changing? And did you also consider including the boundary separation parameter, so A, to see if that changed as well with changes in heart rate?
Let me share. I have a slide with every details of that model, but like I thought there's no way I can have time to talk about it, so I share it right now. Okay. So what we found, okay, so we found a relationship with Z, right? We found that the effect that we found regarding Z was that larger pre-RR. Oh, there are some mistakes here, but listen to me. So larger pre-RR increased starting point Z. So it caused an initial bias. And that makes sense because larger RR means lower heart rate and means more intense sensory intake. Means like there is more reception from outside. So the stimulus is encoded as more intense, or similarly, we expect more intense is equivalent to longer. So that's how it impacts the starting point, the Z. Your second question was about decision thresholds. We did not change the decision threshold. Let me think why. Because it actually, okay, so we didn't have any specific hypotheses about how it changes decision threshold. Because if we think that pre-RR, for example, changes the initial point, it can't change both the starting point and the threshold. They will, like, the model will get unstable if we change both of them as a function of the same measure. That's what I remember from the modeling. And also our hypothesis was stronger about, like, theoretically it made more sense that that pre-RR is more related to the encoding of the stimulus, because decision thresholds makes the response, like if we increase the threshold, it makes the response both more accurate and makes the response time longer. But we didn't have such a hypothesis. We thought heart rate should make a higher heart rate should make the response shorter. Yeah, but so yeah, we looked at the starting point, but not the threshold. Just brief to answer your question. Does it answer your question?
Yes, thank you.
Sure. Oh, you're muted, Effie.
Hi. Can we ask a question? Or?
Yes, sure.
Yeah. Thanks, Saeedeh. It was a very interesting talk. I wondered about the reaction times. So typically, at least for longer intervals, there is an asymmetric shape to the function, such that the shorter stimuli have a longer reaction times. I wonder if that's something you see in your data as well.
No, we didn't.
No,
So we didn't see any relationship between objective duration of the tone and response time.
Mm-hmm.
But we saw a relationship between subjective perception and response time. When the tone was perceived longer, response time was longer.
That is interesting, right? Because, like, if you think about the typical DDM for bisection, like the one from Balchin and Simon, they have this, right, a general trend to decrease reaction time with longer stimuli. But it is for longer, generally longer intervals.
I think the difference is that their stimuli are much longer. So in our model, actually, our model is a bit different from theirs. We assume that the encoding of the stimulus, the entire duration of the stimulus is part of the encoding. It's not the evidence accumulation. So so if we think about it this way, it actually makes sense, because when the stimulus is when the response is long, more evidence needed to be accumulated. So it took longer.
Thanks.
You're muted, Effie.
Okay, sorry, I always forget that. I was asking if there are any more questions. I cannot see any raised hands. If there's any questions in the chat, Martin, can you double check with me? Because I cannot see any question coming in the chat.
There are no questions.
Okay, I have a short one, and if there are other questions, then we can move to the next speaker, if that's okay with everyone. I was wondering if you attribute the behavior of the heart based on the duration intervals, because I saw this comparison at the end. So do you think that the behavior of the heart and its interaction with time perception has to do with the duration of the stimuli or other characteristics? And I'm asking that because I think I really agree with you that the heart is very informative and maybe present a different behavior based on the mechanism that we trigger with our stimuli.
Sorry, what was your last sentence? If it is trigger the...
Yeah, the main question was if you think that the behavior that you observe in the heart has to do
Yeah.
with the
Yeah, I got it.
duration.
Mhm.
Yeah.
Okay, so yes, that's a very good question, actually. I think I don't know if it has to do with the duration, with the time scale, but I think like in a long time scale, heart rate changes for many different reasons, mainly for breathing, but also for many other things. And when we look at it all, so for example, in the long time scale test that I had, something funny, like one of the subjects that I saw that there was a like I felt that this was the experiment is about sexual assault because somebody in the VR environment looked at me in a weird way. Like, you know, there are so many things going on in people's mind in a long time scale that we don't know exactly what's going on with their heart rate changes. So in moments, there could be moments of attention, there could be moments of defense, there could be different moments of like that give rise to the entire duration, to the entire the dynamics of the heart rate. And so I know some people look at HRV, heart rate variability, like they decompose high frequency, low frequency. I think still it is HRV as a trait is good. It shows like it's mainly because of breathing, and it shows how flexible the vagus nerve is. But when we look at it as a state, comparing different conditions, I feel like a lot is missing about what's the underlying cause of these changes in heart rate. There is also another thing that I think is a disadvantage in long time scale, not because it's long, but because of our measurement incapabilities, is that we can't, or it's very hard to separate bias from accuracy in long time scales. And they might have different relationships with the heart, as we saw in short time scales. So in long time scale, we can't have like 200 trials to plot a psychometric function to decompose bias from accuracy. And that way, it's hard to tell which one is it. Is it bias or accuracy, and the effect that we see?
Yeah, and I understand why you introduced the second stimulus with white noise, and you tried to break it down to find the actual cause, let's say, or not the cause, the mechanism, the potential mechanism behind it. That's really interesting, and thank you for introducing the future directions as well.
Thank you.
If there are no more questions, I can move on and introduce Antonio to you. And also, if you come up with questions while we're talking, feel free to email us, and we can forward the question to the speaker and get back to you. So the second speaker for today is Antonio Criscuolo from Maastricht University in the Netherlands. Antonio is just starting his fourth year as a PhD student at Maastricht University under the supervision of Professor Sonia Koch. He studied psychology at the University of Bologna, and he completed his research master studies in cognitive neuroscience at Maastricht University. After his research experiences in the Center for Music and the Brain and Oxford Center for Human Brain Activity, he realized that timing in the brain really matters. So in the BandLab, he combines comparative and translational approach, and he investigates how the brain and bodily rhythms process and predict environmental rhythm. So today, the title of his talk is Sampling the Environment with Body-Brain Rhythms. So the floor is yours, Antonio, and we can move to your presentation.
Hi, do you hear me and you see the screen before I start talking just by myself?
Yes.
Yeah, perfect. So yeah, thanks for inviting. Quite excited to be giving a talk at the Timing Research Forum. I've been following you, throughout this journal club. So I'm happy to be here today. So indeed, it's the fourth year of my PhD, meaning that I'm going toward the end of this journey. But I hope that I'll what you're gonna see today, it's gonna be interesting. So this is the BandLab, and this is Sonia, my PI. I don't know if you noticed, but we are trying to simulate some oscillations here in this picture, and Zan, it's sort of face resetting, whatever is going on. So if you're just interested in hearing more of what's going on in the lab, you can check this QR code. Let's say that the fundamental motive of what goes on in the lab, it's this research line on adapting to dynamic environments. So in the first part of the talk, I'm going to try and convince you that this capability to adapt to dynamic environments relies on our capacity to actually process timing and to predict future events. In the second research line, we want to extend this perspective from studying the brain in isolation to include other bodily physiological signals, and so studying body-brain interactions. And there is also a semi-hidden research line in which we're asking the question of whether these oscillations that we look at the brain are actually real oscillations. But before I start, I want to actually discuss how I started, why I have specific research questions, and how I'm addressing them during my PhD. So I'm a musician that studied psychology. And so I very soon became interested in neuroscience of music. So during my stay at the Music in the Brain, we were investigating how long-term musical training influenced neuroplasticity, and so influencing cognitive functions, as well as neuroanatomical and functional changes in the brain. During my research master's at Maastricht University, I was shortly working with dyslexia. Now, for the sake of the study, what I found extremely interesting during this experience is that reading disfluency are usually treated with rhythmic training. And it seems that these rhythmic training, music classes and so on, improve not only reading fluency, but also phonological processing. Then in Oxford, I was working with people who stammer. Also there, I found extremely interesting that speech disfluencies disappear whenever they are talking along with a metronome, whenever they are tapping a rhythm themselves. Whenever they are singing, they are fluent, but also if they control their breathing in order to speak, they become extremely fluent. So also there was something about timing that was really important. And I started chatting about this with Sonia. So I'm there, Sonia, I think timing and rhythm are very important. It generalizes the different cognitive functions. We should do something about it. And she's like, yeah, well, this is what I built my career on. So I guess we could do something. So I started my PhD. And among the first things that I've been doing was writing a little journal club article in which we discuss with a colleague, Katerina, why this rhythm network in the brain is important for language and speech and why it may actually go beyond. Then we extended this perspective to include influences from the body in the equation. So trying to understand cognition via body-brain interactions. I got a little grant to visit the Max Planck. This was last year. Before the end of my PhD, I'm going to visit Joachim Gross in Münster for a new data collection. But also, what I'm extremely excited about, it's a new initiative, the Body Brain Waves Conference, organized by me and Sonia, and in my hometown. So I'll be talking about this later. But before, I gotta convince you that body-brain is actually a new important topic. But first of all, timing. Yeah, it's important, but timing what? So here I just want to quickly make the point that whenever we are exploring the environment, what we constantly trying to do is extracting the precise timing of events. And not only single events, but we actually also try and understand whether the temporal proximity between adjacent events is actually important in order to understand what goes on in the world. And we do that in the unisensory as well as in the multisensory domain, as in the case of speech. It becomes then clear that in order to transition from chaos to cosmos, we use temporal organization. We process timing. We actually parse inputs in time. We use grouping, segmentation functions, but we also try to predict what comes next. So the first research question for today will be, how do we encode event timing? Very simple setup. We record EEG while people listen to isochronous equitone sequences. These are identical tones interspersed by either 1 or 2 deviant tones. These are just amplitude attenuated, but they are identical in pitch and timing with all the others, and everything happens at 1.5 hertz. So easy setup. And what we're trying to do is understanding whether neural oscillatory activity, as recorded with EEG, reflects the temporal regularity in the acoustic environment. Now, a typical approach to address this question would be to run Fourier analysis. Here, actually, it's a intertrial phase coherence plot. So using the complex spectra from the Fourier analysis. And what you will be searching for is a peak at the stimulation frequency, right? 1.5 hertz, reflecting the frequency of the auditory sequence. And this has usually been interpreted as a measure of internalized timing. What we like to do a little bit more is actually exploring the time course of neurooscillatory activity, trying to understand whether we actually track timing, if there is also any type of anticipatory behavior in this oscillatory activity. And we try to see whether the pre-stimulus phase is actually coherent within a sequence. So we would use circular statistics, in this case, mean vector length, at the single sequence. So this is single subject, single trial level type of analysis. And we compare these mean vector length distributions against random distribution. Now, this single trial approach also allows us to look at intra and inter-individual differences. So you may notice that part of this distribution actually mirrors what happens with random phase angles. And some other times, instead, we will say that you are tracking. Now, such an easy setup has been used to perform comparative and translational studies. So we have a sample of macaque monkeys. We have stroke patients with lesions in the basal ganglia or cerebellum, and we have an healthy aging population. So this is what the overview of the results would look like, with very interesting patterns of similarities and dissimilarities across species and experimental groups. Unpublished work for now, but hopefully you'll see it out soon. The second question, how do we go beyond? Obviously, we not only process single event timings, but for processing rhythms, we need to extract temporal regularities, and we need to go much beyond. Now, let me see if I can click on this video. Ah, perfect. And there is a bit of advertisements. Well, so yeah, in order to process the rhythms, we need to extract temporal regularity. What we do next, and this elephant does it very nicely, it's synchronizing behavior with rhythms. And we do so by making subdivisions of what we listen to. We make grouping, we make accentuations, but also we try to make temporal predictions in order to behave also flexibly, so varying behavior over time. But how do we do so? So yeah, I hope that was a funny video, something new. Interested in sampling the acoustic environment, we are saying we don't do it tone by tone, otherwise we will be very boring dancers, and probably the elephant, you know, it's much nicer than us. So accentuations, how do we study these accentuations in the brain? Now the idea was that we look at this isochronous sequence as we have looked at it before, and we simply try to model accentuation patterns. So this will be a tick-tock, strong, weak type of accentuations. This will be a strong, weak, weak, strong, weak, weak accent, so pulse-like, and this will be no accentuations at all. And we look at time-locked responses in the beta band informed by literature. With such approach, single subject, single trial, as usual, we can get a distribution of preferences for accentuation types. We can get a distribution by zooming into binary and ternary trials. So to look at where these accents are placed along the sequence. We can also create pairwise amplitude difference matrix. These are tone-locked beta responses for strong, weak events. So this will be the position along the auditory sequence. And then we can extract some metrics for how similar this strong position are, or the weak position are, or how dissimilar the responses to strong versus weak, or weak versus strong are. And next, after having confirmed that these standard tones are processed with binary accent in those trials being classified as binary, we also look at deviant tones. And it turns out that these deviant tones are differently processed when they follow on strong and weak positions, but they're identical. Now, it's also nice that we replicated these in 2 macaque monkeys, getting very similar patterns here, although these matrix look a little bit more scattered than the first one. But I want to zoom here into single subject recordings in the standard tones and strong and weak position. You can actually realize that what happens there, it's really a pattern of accentuation and suppression for strong and weak position of identical tones, and in the monkeys as well. And keep in mind, there were no instructions. This is just passive listening, and we interpret it as a way of sampling the environment at a different time scale. So in this case, superimposing accentuations with the beta band. Then the third questions, are neural oscillations really oscillations? So we've been using this term oscillatory dynamics. We study entrainment, synchronization, phenomena, and so on. But before doing that, we need to check a few things. So we need to check that what we look at is actually a self-sustained oscillator. This oscillation should pre-exist and outlast the stimulation period, for example, our auditory sequences. And this oscillation should be able to tune in and out of environmental rhythms, should show flexibility. And this flexibility is what would allow adaptation to dynamic environments, and may also be informative when we look at individual differences and clinical conditions. So how do we are trying to do that? Again, single subject, single trial estimations. This is what we've been seeing in the first part of the talk, now complemented with something more, time frequency analysis, but also instantaneous frequency. So this will be dynamic changes in the frequency content of oscillations. And here what we do is extracting metrics and distribution for what happens during the auditory sequences, but also what happens before and what happens afterwards. So quantifying mean power, mean frequency, but also entropy changes over time. And obviously, this is a single trial subject and channel estimation, so a few thousand figures sort of summarized in these plots. Here, trying to disentangle cortical subcortical contribution with cerebellar and basal ganglia patients. So with this, I hope I sort of convince you that neural activity dynamically samples and predicts timing of events in the acoustic environment. Where we are neglecting, however, is the body, so physiological signals from the body. And in the second research line, I still have quite some time, new research line on body-brain interactions. At some point, we realized these physiological signals from the body may be a neglected source of information, and it may be very interesting studying whether they inform human cognition at all. Now, in this talk, I will simply focus on breathing for the sake of time. Heart was covered in the previous talk, and but just keep in mind that there are many other bodily signals, for example, gastrointestinal rhythms, circadian rhythms, the eye, and so on, which are very, very interesting. And yeah, we try and summarize the literature in these two papers. And here there is a list of references that I'm going to use in next slides. So focusing on behavior first, early studies showing that the finger movement of pianists would couple with breathing patterns. There is then evidence that breathing influences action, from walking to speaking to music playing, but also reaction times and precision. There is evidence that breathing is influenced by sensory input. For example, there is evidence for respiratory entrainment to music, but also that is influenced by psychological states, for example, states of anxiety and stress. The top-down control of breathing can also influence emotional control and cognitive functioning. Moving to neural data, this study from 2018 shows that if you continuously monitor brain activity and breathing patterns over time, you can quantify coherence, but also this breathing to brain coherence is modulated with breathing rates, so moving from fast breathing to natural breathing. And these modulatory effects of brain breathing coherence, it's found across widespread cortical subcortical networks. Now, more recent study from Daniel and Joachim further characterizes this respiration modulated brain oscillatory network. Further shows that it partially overlaps with well-known existing networks like the default mode network and attentional networks, but they also zoom into inter-individual variabilities and frequency variability of tuning patterns. They also remind us in the introduction that studying breathing is important because we all breathe. Yeah, I found a nice way of starting a paper. You know, usually you're stuck with the beginning, but this is, yeah, good idea. Moving to motor preparation, here a study showing that the brain readiness potential, this negative slope before action performance, seems to cluster in the inspiration phase of the breathing cycle as a mechanism of preparation to action. And the last paper that I want to show you, this is invasive recording in rats. Here they show that if you keep on monitoring these brain breathing interactions over brain states from sleep, running, exploration, and no matter what you use to monitor these interactions, so correlation, coherence, phase shifts, and so on, you keep on observing very high patterns of correlated activity. And they also use Granger causality, which gives you also a directionality of influence. Keep this in mind because you may see it in next slides. So, right, it was clear that there was enough evidence out there that the body, the signals from the body are quite meaningful for information processing in general. And so what we tried to do in this paper, it's after a brief review of the literature, is trying to convince the reader that we should take a perspective on how a body-brain dynamic system may inform human cognition and any type of observable behavior. So using this partially neglected source of information of bodily signal and use them to inform our examinations of brain and inter-individual differences. The starting point is simply the not disregarding these signals that we pick up from the body and that we usually take out with artifact rejection procedure. And we want to actually describe a system that shares and exchanges information with the environment and delivers behavior via transformation processes. Now, a couple of working hypotheses in order to move to experiments. We want to characterize the dynamics of these signals fluctuating over time. And we think that no matter what metric you may use, what we may observe if you're doing any type of phase lock-in analysis, transfer entropy, Granger causality, or whatever, is at least two states, a decoupled and a coupled state. So these body-brain signals are not always synchronized over time, but they try to achieve synchronization whenever contingencies require. And now the sweet spot lies in between. There are some state transition. So there are moments in which either of these signals may trigger some state changes, either by speeding up or triggering a phase reset or any other mechanism which we should study in order to understand how and why we use these body-brain interactions for any type of behavior that we may be interested in. A few examples starting with breathing, because we're all experts in breathing control. After all, it's among those few bodily signals which we can control top down, and we do so for any type of activities, from the speaking, playing instruments, running, and so on. We know that there are different breathing patterns according to which behavior we should perform, and we implicitly control top-down breathing in order to improve even cognitive performance or to direct attention to the environment. But now the question is how this top-down control of breathing also triggers state transition in the body-brain dynamic system. The other angle then it's looking at the heart, for example. This cannot be top-down controlled, but is actually modulated by controlling breathing. What we know of the heart is that, as we also heard in the previous talk, that it responds very swiftly to changes in the environment. For example, when an unexpected event occur, for example, a cat jumping at you. And also here, it's interesting to study what happens in the state transition and how this is reflected in the brain. So there is a pre-registered study sampling the environment with body-brain rhythms in which we're using mobile EEG. This is the Muse headband. Actually, it's two of them because with one of the two, we are monitoring activity from the body. Yeah, experimental setup, maybe I should be faster. So I will just say that we have a little bit of everything from rest to walking, speaking, and listening conditions. I just want to show you what the approach is, but we are in the early stages of the piloting. So I don't have too much to show, but some preliminary analysis. So this is a Muse headband placed on the chest during resting states. I want to show you that you can get some very nice heart and breathing data. It also works fine with walking, and also look at the length of these recordings. These are very short, and you get even a individual walking rate, breathing rate, and so on. But we also are examining the time course and quantifying Granger causality. So directional influence from breathing to heart to breathing compared to shuffle distribution, and using this Granger causality metric with a network perspective. So directionality of influence and strength of influence. Lastly, and I'm really at the end of the slides, I'm 1 minute later. We want to build an individual holistic body-brain frequency architecture. What this means is that we start feeding information in this architecture of individual heart rates, respiratory rates, so body and brain activity, and we want to try and test whether there is any link with individual rhythmic behaviors, spontaneous tapping, spontaneous walking, speaking, but also preferences for listening specific music, for example. Obviously, we're interested not only in peak rates, but in variability, so in the dynamics of body-brain interactions. And obviously, we want to transfer this knowledge to clinical studies. So with this, timing. Oscillations in the body-brain dynamic system may help us understand how we coordinate perception and action by encoding temporal regularities in the environment, anticipating future events. And what we try to do is having an individual and holistic perspective. We're moving towards neurocognitive profiling, and we want to try and understand how we adapt to dynamic environments by anticipating future events. With this, I want to thank my lab, very exciting environment. I want to thank collaborators and a couple of funding bodies that supported us. Yeah, also it's a nice lab. From time to time we gather also to cook together. Here I'm teaching the guys how to make tagliatelle at home. Some other times it's lasagna, gnocchi, or whatever you may. This is Aperol Spritz as well. And so, yeah, good pasta, good chats. There is lots of projects going on. So just check our website, get in touch if you're interested. And Body Brain Waves Conference. I'm very happy to just show you the preliminary plan of this conference, which is going to happen in June. It's preliminary in the sense that it took me a while to get in touch with the local politicians and so on, but it's happening. So also here, stay tuned if you're interested in this topic, if you want to contribute or if you want to support us in any way. And I hope I will see you, soon, either at this conference or at the Timing Research Forum conference. So thank you for attention.
Thank you so much. That was a really informative presentation, and one comment from my side about the last project you introduced. That's a very, in a good way, ambitious project, and I'm really looking forward to see the outcome. Let us know about the details on the conference, and we can definitely include it in our newsletter so we can spread the word. Let's see if we have questions.
I'll leave it on just in case.
Oh yeah, definitely.
It's just a brochure, but you will find my contacts. Yeah.
Okay, I cannot see any questions in the chat. I have a short question about the last project. Are you including circadian rhythms as well in your study, or only breathing part and stuff like that? Because it sounds fascinating. If you, I was about to ask the way that you're planning to induce that.
Yeah, no, circadian rhythms is in the conceptualization, but not really in the experimental part. Also because we are obviously collecting data from the student population here, and I wouldn't rely on students' circadian rhythms with heavy schedule of partying and studying, all mixed up with good alcohol, and in the Netherlands more. So yeah, that's, for now, it's out of the equation, although is in the conceptualization and in this review. But all the other bodily measures, yeah, for sure. So we are also actually thinking of including skin conductance, gastrointestinal recordings with additional electrodes. But yeah, we are in the piloting phase. So now I was taking care of also setting the analysis pipeline so that I can hand it to students and, do some heavy data collection, and then having everything analyzed ASAP. Also because I should finish this year. And so there is the writing of the thesis and grants for afterwards and so on. But yeah, I think it's a tough but good type of field, this body-brain interactions.
Yeah, that would be really interesting. I was running some studies in circadian rhythms. Definitely, I can see the difficulty in the early adulthood because of the messy schedule. But definitely in older population, that would be an idea. Might be to have a watch, let's say, for a week, and you can collect continuous data and put them in a model, for example, if you are gathering other measures as well. I'm really interested. I'm looking forward to it.
Yeah, we actually do have sleep recordings. I probably forgot to mention. I actually couldn't put it in the slides, but I've also analyzed some links between sleep rhythms and behavioral rhythms, which seems to be interesting. So yeah, actually I could use that for circadian rhythms. That's a good idea. Thanks.
Because it can capture really small motion during the night, and there are a lot of I wouldn't advertise it right now, but there are a lot of equipment you can look and be quite accurate for research and sleep architecture. You can have a full profile for a whole week, for example, so you can see before any experimentation or afterwards.
It's a good idea. Thanks.
Oh, you're very welcome. That's why we're here. We exchange ideas and experiences.
That's cool.
Okay, if there are no more questions, don't forget to keep an eye on our post newsletter. We will have all the up-to-date details about our conference. And I must say that in the next talk is going to be given by Anne Geers from University of Strasbourg on 22nd of February. So stay tuned and we hope to see you soon. Thank you for attending the talks.
Bye, everyone. Thanks.
Bye.